Hi, it's Alex MacKenzie. I'm delighted, if somewhat puzzled, to be asked to be the moderator, given my organizational skills, but there you go. So I started out back in the almost the late 80s, working on a disease called spinal muscular atrophy, SMA. And it was roughly 1995 or so, 96, I was invited to the first family based organization, very analogous to this, to get together and a gathering not dissimilar, although 30 years ago now, not dissimilar. And I just have to say, it over the ensuing 2 decades, it led to the formulation of not one but 2 effective therapies for spinal muscular atrophy. A drug known as nusinersen, which is an antisense oligonucleotide drug, and an AAV9-based gene therapy out of Brian Casper's work. And while that was ultimately funded by NIH and pharma in the later stages, it really started with a family organization very much like we have today. So there is hope, and it's going to be much more telescoped for your path than it was for our path. The AAV9 is there. We're going to hear later on about the AAV9 utilization, et cetera. So it encourages me at the sort of Joe Biden stage of my career to be dragged into this, to be involved at this level, because I'm very optimistic about how things will work out, and but it is very energizing and helps us researchers, get things going and really focus our minds, which I always need, I have to say. But so I want to just open with those remarks and thank you, Mara and Sprina and others for all the work that you've done. So I've lost the thing of the agenda. I think we're going to start off with Alex, the smarter, younger Alex, as I say, who is I first met Alex about 5 years ago. I was attending pediatrician at the Children's Hospital of Eastern Ontario, and he was sitting on the bed, Cedric's bed, sort of interacting with Cedric, and you guys were just a patient under me. I'm just do general pediatrics, and since that time, Alex, I've got to know the Gervais and Cedric, and it really altered the course of research in my laboratory, as Gerardo Medina will tell you later. But Alex went on. He was just an undergraduate at Carleton at that stage, at university, but he went on to become a genetic counselor and really a content expert in DLG4, and as well having his own independent career. He was a very central part in something called Think Rare, which is a way of mining the electronic medical health records for rare diseases. As you people know, there's this diagnostic odyssey where you go from this disease and all its puzzling manifestations to actually getting the diagnosis. Alex is working on, in addition to the DLG4 aspects, a way of going into an electronic medical health record and identifying individuals with a rare disease that need genetic testing. And it's the first sort of platform like that in the world internationally, and he really was a key part of it. So he really is an impressive young man. He's going to just give us sort of DLG4-SHINE 101 course right now, and I don't know if he's on the line. We can switch out. I'll shut up and we'll replace me with another Alex. Okay, thanks very much.
Perfect. Okay. Well, thank you so much, Dr. MacKenzie, for the lovely intro. And I'm not sure if people can see me, but I am wearing my DLG4-SHINE shirt today in my bright orange. And if you ask Mara any opportunity to wear orange, I will gladly take it. But I am really apologetic that I can't be in person to be seeing all of you today, meeting some of the families that I've gotten to know virtually over the years, and some of the new families, and even seeing some of the numbers that Mara presented, 200 families, and it's been 3.5 years now of the foundation, and I can remember working as a respite worker with Meg Faust and Dave and Cedric. It was always just a thought that this could happen. Now seeing where it is today, it's so truly impressive, and I'm really excited to be a part of it. And The goal of my presentation today is really just to be a very brief overview of some of the genetic concepts of how they relate to DLG4-SHINE syndrome, and hopefully provide a really good foundation and primer for some of the more complex talks that might be coming later on today about the therapies and how the proteins and genes function. So I'll keep it very brief as mentioned. I'll talk about chromosomes, genes, proteins, the different types of mutations. I know Sabrina touched on a couple of those, the inheritance pattern of this condition, and a question that a lot of families come up with is that of mosaicism. So I thought it might be important to touch on that a little bit as well. So when it comes to our chromosomes and our genes and our proteins, we're really talking about those building blocks of human life. And for many of you, you may have seen this picture before. It's used by a lot of genetic counselors and geneticists when talking about how we go from our cells and our chromosomes all the way down to our protein. And you may have seen this maybe in your biology classes in high school or depending on your background, but essentially our bodies are made up of millions and millions of cells. We have skin cells called fibroblasts, we have brain cells called neurons, all of these cells. And inside of each and every cell, we have 46 chromosomes. These chromosomes are large packages of our DNA. And we have 46, 23 that come from our mom and 23 that come from our dad. And if we were to pull these chromosomes apart and string them out just like this, we would have a very long string that would be our DNA, the instructions or the building blocks for telling our bodies and organs how to develop and how to function. And if we think of that long string and we think of thousands of beads along that string, we can think of the beads as our genes, as these little pieces that are put together to make up our DNA inside of the chromosomes. And we have about 20 to 25,000 genes. We're very complex beings. We need a lot of genes to build a lot of complex organs and have our cells operate the way that we need to. And all of our genes are spelt with a very specific code so that they can be read to create very specific proteins that have jobs in the body. Now, the coding of the genes is made up of 4 letters, A, T, G, and C, and these are the nucleotides that make up that code. And the spelling of those genes, the way that those nucleotides are put into place, is very, very important for how these proteins are pieced together that have the jobs throughout the body, whether it be making sure that our cells are communicating or making sure that our skin cells are reproducing, all of those different things. So if we were to zoom out and look at all of those 46 chromosomes, this is kind of what it would look like. They'd be a lot more scattered, and this is a lot more organized view. But as you can see, we have 2 copies of every chromosome, so a pair of each, all the way to 22, and then we have our sex chromosomes as well. XY for male and XX for female. The chromosome that I really want to focus on today, though, is chromosome 17, as that's where the DLG4 gene is located. And given that we have a chromosome that comes from mom and a chromosome that comes from dad, we have a maternal copy of the DLG4 gene, and we have a paternal copy of the DLG4 gene. All of us have 2 copies of this gene. And the DLG4 gene, just like any other gene, has a very specific code. Now, this is not exactly the code here. This is just an example, and thank you, ChatGPT, for putting this together. But as you can see, we have A, T, G, and C making up the code of the DLG4 coding all the way through. And the coding of that gene, as I mentioned before, in all the genes is really important because when we look up here at the top right, these letters in the DNA and in the genes are grouped together in triplets, in 3s. And the way that those groupings, those triplets are spelt is really, really important because that spelling helps to make the pieces of the protein called the amino acids. And the way that the little triplets are spelt make different types of amino acids that build this protein to the size and shape and function that we need it to be. Now, when we talk about the DLG4 gene, when the code is read for the DLG4 gene and it's making its protein, the protein that it's making is called PSD95 or postsynaptic density 95. And this is a really important protein because it acts as a scaffold. It brings together and organizes lots and lots of proteins at important areas in the cells. specifically in our brain cells, so in our neurons, and the connections of all of those neurons. So it has a really important role. So what happens when there's a spelling difference in the coding of those genes? And I like to try and at this point provide a little bit of an analogy or a metaphor of, if we think of our chromosomes kind of like chapters in a book, our genes are like the sentences in the chapter. And when we're reading a book, when we're reading a sentence, we expect it to be spelled in a very specific way so that we can read it and understand it. Now, if there's a change in, let's say, one of the words, so in the sentence, the car was red, if the word red has an E replaced by a D, we no longer know what that means. We don't know if that's supposed to be red. We're not really quite sure. And again, if we take the word car out of this sentence, all we have is the was red. And again, we're not reading it the way that we would expect. And these are what we call mutations. They're like misspelled words or disruptions to a sentence. And we'll go through what all of these different types of genetic mutations look like and what they do to the protein. So the first type of mutation is called a missense mutation. And you can see again here we have our little triplets of the DNA coding along here. And this would be what we call our normal sequence or our reference. And many, many years ago, all of our genes were looked into to try and build a reference so we know exactly how this spelling should be for each gene. And we can see that these groups here, they make the right amino acids to build the protein. And for here, GCA should make an amino acid called alanine. However, in this case, for the abnormal sequence, we're seeing that this letter G is being substituted for letter C. And this is changing the outcome of that amino acid. So from an alanine to a proline. So if we look to the right here, we can see that wild type, which is just a fancy word for normal, when the gene is spelt normally, this creates the right normal protein that functions and folds exactly how we would expect. However, if there's an altered codon, so here we have this codon or this group of 3 letters that changes, it changes the one amino acid, and this can actually impact the way that that protein is able to fold or the ability for it to interact with other proteins. So it can change the way that the protein functions, and this can sometimes cause DLG4-SHINE syndrome. Similarly, we can have nonsense mutations, sometimes also called stop-gain mutations. It's similar to a missense mutation, except instead of a spelling change causing a different amino acid to be made, the spelling change here from A to T is actually causing a premature stop codon. And what this means is that instead of the stop codon, so this specific sequence here, being all the way at the end when we want the protein to be finished, it's happening earlier because this change is telling the protein to stop producing. It's causing the amino acids to stop making the protein. So we end up with a much shorter protein because the stop codon is a lot earlier. And that protein is not usable. It degradates, and it's unfortunately not usable and can cause the condition. For a frame shift mutation, this is a little bit different. We don't have a substitution of a letter. Instead, what we have is either an insertion or a deletion of a letter or multiple letters. So here we have this G, which is being deleted or taken away. And what happens is the letters here are shifting. So if we think of this as the reading frame, the frame is shifting over, so a frame shift. And if we look at the consequence of what this means, as we shift all of these over, the groupings become all different for making their amino acids. So we have many different amino acids, not just one different, and all often will lead to a premature stop codon, which again will lead to a smaller protein that will degrade. And lastly, one of the more rare types of mutations, although Sabrina was just mentioning on that report that she was showing, we can also see splice site mutations. So here, if we think of this top part, exon 1, 2, and 3 in this string here as the gene, as a DLG4 gene, all of our genes have these really important coding areas called the exons. This is where we get those groupings of letters that are really important for making those amino acids and protein. However, in between these exons, we also have what we call these non-coding regions, although they do have a code in them, but they are not used to help make those amino acids and proteins. So what we need to do is we need to splice these away. And there's splicing mechanisms that help take these away so that we get this block of exon 1, 2, and 3 together so that we can read it and make the amino acids and make the protein. But sometimes if there is a change in the spelling of the intron of this piece in between the exons, this can cause abnormal splicing, where sometimes an exon can get skipped. So we end up with exon 1 and exon 3 without 2. And therefore, we're missing a lot of those triplets of letters, and we're missing some of the amino acids being made. Or sometimes we can have retention of the intron, which can also cause issues with how the protein is being made. So turning more towards the inheritance, so how these genetic mutations arise in families, how they're being passed down from generations, for example. When we talk about DLG4-SHINE syndrome, it's inherited in an autosomal dominant fashion. So what this means, and I'll go into more detail about how we see this condition more commonly, but from a general standpoint, if an individual has DLG4-SHINE syndrome, they have 2 DLG4 genes, similar to everybody in this picture, but one of their DLG4 genes has a genetic mutation, causing the gene not to work and the protein to be degraded or not functional. Every time this individual were to have offspring or a child, there would be 50% chance of passing down that gene with the genetic mutation. That's because this individual could pass down their copy with the genetic change or their normal copy. And we know that their partner is going to pass down a normal copy. So there's a 50% chance. And if we think back to we have our 2 chromosome 17s, we have our maternal copy and our paternal copy of DLG4. The mutation could be on the paternal copy, could be on the maternal copy, and it could be something that we call de novo. So for many of you and most families with DLG4-SHINE syndrome, this is typically the type of pedigree or family tree that we might see, where both parents do not have the condition, but they have a child who has DLG4-SHINE syndrome. And this child would either have a genetic mutation on their paternal copy or on the maternal copy, but it was not something that was necessarily present in all of the cells of the parent. And this is where I really want to talk more about mosaicism and how these de novo mutations occur and why it's still possible that if the parents are not affected, how this can reoccur in families. So just a very brief kind of quote or description of what we call germline mosaicism. This, also called gonadal mosaicism, is a type of mosaicism where one set of genetic information is found specifically within the egg or sperm cells. And this can be seen, so mosaicism as a whole just means that there may be some information in the DNA that's different in some cells compared to others. So we might see a genetic mutation in some of our cells, and in other cells, we might see the normal spelling of the gene. And this image here at first will seem really, really confusing and a lot of information at once, but I wanted to break it down to try and give a better understanding of how these de novo mutations occur and what we know about this information right now. So what this image here shows are all the different types of ways that a mutation can occur in the sense of a de novo mutation. And when we look in the literature, what are the proportion of cases that we see? And can we determine the recurrence risk based on this? So the first one, and based on the literature that we know is the most common, is what we would call a paternal one-off mutation. And if you look up here, we have a child with the condition. But when we look at when the egg, this large circle here, is being inseminated by the sperm, you can see that only one sperm cell is purple. And that means that there's a one-off mutation in one of the sperm cell where all of the other sperm cells are expected to be unaffected or not have that genetic mutation. And it happens to be that one sperm cell that inseminates the egg and leads to the pregnancy. In this case, the recurrence risk is considered negligible because we are assuming that the rest of the sperm cells are unaffected. Now, for most individuals, and I know at my center at CHIO, I work in prenatal genetics, and unfortunately, a big question we get is, can we test all the sperm? And it's not something that we offer at our center, but I know there are other places that can test the sperm and try and build an estimate of, was it a one-off or are other sperm affected? And that's where we get over to our gonadal mosaicism or paternal gonadal mosaicism. And the difference here is that perhaps we are seeing not just one sperm cell that has the genetic mutation, but several. Not all, but perhaps several. But we're still seeing that only the gonadal cells, so only the sperm cells, are being affected with this genetic mutation. And we're only seeing these in about 2% of cases based on the literature. And the recurrence risk is really hard to quantify. It could be anywhere from 1% if only a small amount of sperm cells have this mutation, all the way to 50% if a very large amount of these sperm cells are affected. If we move over to mixed mosaicism, it's very similar to gonadal mosaicism, where some of the sperm cell or most of the sperm cells have the genetic mutation. Here we might see sperm cells that have the genetic mutation, but also some other cells in the father that may have this genetic mutation, but not all of the father cells. So we're seeing some cells, but not all. And this can still lead to an increased risk, anywhere from 1 to 50%, depending on how many of the sperm cell have the genetic mutation. Similarly, with if it's a maternal mutation or coming from maternal origin, we can see one oocyte or one egg that has a genetic mutation where the recurrence risk is negligible. We could see several egg cells that have the genetic mutation where the risk could be 1 to 50%, or again, mixed maternal mosaicism where some of the mother cells may have the genetic mutation, including some of the egg cells. And lastly, sometimes we can see a post-zygotic mutation where none of the sperm cells or none of the egg cells had this genetic mutation. It occurs after the egg has met the cell, as the cells are dividing. And the extent to these cells that are affected with this genetic mutation really depends on when in cell division this type of mutation started. If it's very late, after many cells have already divided, it may only affect a small subset of cells. If it's very early on, it may affect more cells. So I know it can be a bit of a confusing concept, but I do know that it's a very common question that's asked. And in front of you today, whether you're there in person or over Zoom, you have some of the best researchers really in the world, in my opinion, those who are extreme experts on not just basic genetic concepts in DLG4, but across all different genes, all different conditions. So definitely pick their brains, ask them more questions about these. And you're more than welcome to reach out to me at any time. Please just ask Mara or anybody there for my email. And I'm happy to be a resource or advocate for any of you at any time if I can. And I'll just thank you all again for having me today, and enjoy the day, and all the best to you.
Great stuff.